Yes, good morning to all. So last we were talking about microbial reproduction, sexual and asexual, right? What is meant by asexual? Which mode is used in bacteria? Huh? Yeah, it's a binary fission. Very good. For the sexual reproduction, which mode is used? Binary fission, asexual reproduction, right? Sexual reproduction, which mode is used? Or the way? Yeah, it's conjugation, conjugation breach. Pili-like structure is get extended. Breach formation for the transfer of the DNA. So in case of reproduction, it is nothing but the transfer of the genetic material from one to another individual between two cells is just considered as a reproduction. It may be get acquired, so we are going to look for the other mode of reproduction or the DNA transfer. There, either from the environment or the others are participating into it. Not actual cell is doing the genetic material, some others. So now we are looking for the phages. What are the phages? Bacteriophage that infect the virus that infect the bacteria are called a bacteriophage. Generally, maybe you have learned about the T4 family or the muphage. Certain phages are there. Some maybe you have learned or heard in previous. M13, have you heard about M13 phage? Mu, T4 at least, T phage. So no need to worry about it, but I can show some pictures related to T4. So transfer of the genetic material via the viruses is going to take place. It's the method called the transduction. In the whole question can be asked as a asexual reproduction, sexual, or the DNA transfer, other mode, or the mode of reproduction in 8 marks, 6 marks, 4 marks, any pattern. Okay, in any pattern in exams can be asked. So there are in transductions, the two different ways are there. One is a generalized and another is a specialized. What's generally going to happen into it? We are going to see them one by one. Here you can see it is a landing platform. It's one bacteria. One is a phage. Here you can say this one is a T phage. Maybe you have seen in your previous somewhere, maybe in the 12th. I'm not so sure. If it is not, you are going to learn first time, then also not a problem. So what are the basic sections are there into phage? Each phage cannot affect or infect every sort of the bacteria. It is like a viruses, coronavirus. People were thinking, is it able to infect any other mammals or not? That is like a cow, cat, or monkey. They cannot because of their receptor availability. Most important for every sort of the interaction. And like you are learning into your some chapters also for communications and others. For every interaction, whether it's a verbal or a receptor based or anything is there, there may be one is a communicator, one is a receiver. This two system has to be established in any sort of the things, whether it's a material exchange or a sound exchange. So it is like a physical, chemical, or biological. In each category, one is a start to send something, another is start to receive. This mechanism very particular and precise. So here, the bacteriophage do have some viral genome. There may be a DNA virus and RNA virus. Here we are looking for the bacteriophage, and we are looking as they are with the DNA as their genetic material. Generally, in case of animal, Mainly it has been observed they are the DNA based viruses. In the plant it is the RNA based virus. So during the evolution they have separated, but yes, there is a exceptions always there. DNA containing RNA containing. What is the corona genetic material? It's the DNA or RNA? DNA or RNA? Anybody knows? Coronavirus. You are from the microbiology. At least we have to know whether it's RNA. Okay. So RNA virus. So always there is always an exceptions. So when it is moving, jumping, so it is one targeted. So mainly it's going to affect on the lungs. Spike protein. For the corona, we are knowing that spike protein only been accepted for their infection. In first initially, it has been observed that teen guys up to 14 or 18, they are not going to get infected. The reason is what? Spike protein development in a lungs, it's not mature. In a teen guys, whether it's a girl or boy, throughout the world, it has been observed that spike protein were not mature for their interaction. Okay, so spike proteins, where it is available, and it is going to infect, and that interactions was not. But afterward, it has been observed that viruses get evolved, and during the evolution, now they start to infect to even the children also. Reason is what? The main machinery part for the interaction was there. They have devised themselves in different mode now. So they can infect even to the lungs of a child even. Earlier it was not. So how they have evolved, that is a total mystery. Okay, so they are looking when they are infecting, getting infected, they want to survive into the just in a child, but they cannot sometimes. So they have evolved surrounding area. From that, according to surrounding area, they have evolved themselves to infect them. So otherwise, the child were just carrier. Now they are became a, they for them host also. So here it's the host pathogen interaction. So here first thing is what? It's the landing. Viral particle is going to land onto the bacteria. Next step is what? They will introduce. It is like a transfer of the genetic material to the bacteria. Next step. Followed by, yes. Now it will, in the third step, second to third step, that viral genome will going to establish certain proteins and it is going to disintegrate the bacterial genome. Genetic material of the bacteria going to get disintegrated, and virus is trying to cover the thing is what? To utilize genetic material of the bacteria. So within the pouch, it is like that. You are sitting here, one virus has came, okay, or the some person has came, and what is he has done? He fully with arm, so he is going to kill the person, right? Or if it is not a killing, he may snatch certain utensil or the products or the material from. So it is the virus going to do here. And genetic material, that is a totally two things is required for the protein. One is their protein code and another is the genetic material. Only two things is required for the virus. So it is going to use the genetic material of the bacteria. It is disintegrated in a fragmented. So all the nucleotides are available. What it will do? It will use the replicative mode to convert their genetic material, copy, utilizing the chromosomal material of a bacteria. And it will start to pack. During this time, transcription and translations are also going together for the protein code development. Surrounding. Nothing else. It is a cache or it is a surrounding pack. It's going to packing material. What is going to pack? Just a genetic material. Just two things is required. So it will is going to infect the bacteria. It will arrest all the machinery of the bacteria. Protein synthesis, everything is in control. It is like a country. It is been taken by the someone. Like right now we are looking for the Taliban. Okay, it is covered on the Afghanistan. So people are there, but it is in control of some others. So all it is in control of some other guys. So bacteria are now in control of bacteriophage. So bacteriophage, what they are going to do? They are looking to for multiply, nothing else. So multiplications will start. In this process, sometimes you may observe here it is a red color. It is a viral genome. By mistakes, it has been observed. It is a rare phenomena, not always, but by chance it has been observed that during packing, bacteriophage, that protein code is going to take the some fragment of the bacterial genome. Generally, it is going to pack what? Viral genome, red color. But sometimes it has been observed that some phages, it is a percentage, you can say, consider like a 0.001. Frequency is very less, but sometimes by chance this happen that it is going to cover a phage material is going to cover, that is a protein called a bacterial genome. Okay, now the next step is what? It is going to burst the cell. Bacteriophage is their number. If one bacteriophage is infecting, it is estimated, or it is depend upon the phages also, about thousands of phage will release out of one bacteria. So out of 1000, maybe 1 or 2 bacteriophage only do have the bacterial genome along with them. Now what they will look, they will going to infect next to them. Now they will look. Here I have shown the red to blue color. Genomic material is a blue color. Means what? The phage, by chance, that a bacteriophage protein code has taken up the material of the bacterial genome. It should be from the viral, but the packaging is done for worm. Bacteriophage has done what? Bacterial genome has taken up. And machinery is nothing else. It is like a vehicle. Okay, driver is going. It has to take their persons, recognized persons, right? But by chance, what happened? Someone has opened the car and start to sit. So driver is only knowing that someone is sitting, I have to move. It's not going to recognize or not going to identify that who has sit over there. It is the same phenomena here. That bacteria has a bacteriophage has taken the genetic material from the bacterial part. Now driver has moved to next place. It has infected next bacteriophage. What going to step is it going to inject as it was in previous. Now this one is a donor bacterial DNA. Now the phenomena is going to change in this place. Generally, what's happening when the phage is infecting, that DNA material is going to start replicate and arrest the bacterial genome. Bacterial, all the machinery were arrested. DNA replications, transcriptions, translations were arrested, and it start to grow themselves. But here, something has happened differently. Now this genetic material is came outside, and it is similar to the previous to nearby one. The first bacterial was there, and this one is a similar map. So what going to happen in next stage? That is a recombination is going to take place. If it is more or less similar to it, chances are there that a genetic material came out of it. It is having the more or less same frequency. So it is going to replace. Recombination is going to take place, and it will exchange. Sometimes it is going to fully integrated. It may be exchange, or sometimes directly is going to integrate it within it. So there is a chance that the next bacteria is going to get new genes. Maybe it is antibiotic resistance or sensitivity. Anything could be possible. Something is gifted to them. During that time, if someone is coming, one has to move. It may be selective or elective. If I'm coming and I'm going to put one student and I'm just randomly taking someone to move out, or purposefully or selectively it may happen that you or someone has moved out. So it is both the possibilities are here. So donor DNA is going to get fit into the recipient, and this is the mode of a transfer genetic material. Now this virus particle is going to lie themselves there only. It's not going to move, but it has not able to disturb the bacterial genome, because if the viral particle has taken the viral genome, then it has arrested them and the phage productions may be. Now it is not there. So this bacteria is going to integrate with them. So this is the basic transductions method. general transcriptions method where phage is participating into DNA transfer from one to another bacterial cell. So it's called the other mode of reproduction. Previously, what we have seen in a sexual reproduction, some F material was moving. Plasmid was moving to one to another via pilus. In asexual reproductions, there's the division. Third case we have seen there, bacteriophages is participating into material transfer. Even yesterday we have seen about the transformation. So in case of transformations also, it is from the environment, either plasmid or DNA fragment is taken up. Clear till this? I have one small documentary into it. Let's see about it. It's about 1 or 2 minutes. So afterwards,
in generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
The last step what they are telling is that now the new bacterial cell has adapted new genetic material, and then after binary fission is goes on. So, but by chance, what happened that genetic material has been transferred, and this may lead to positive or negative effect. Both the possibilities are there, or sometimes it is going to replace the place or going to integrate. So size of the genome of the bacteria is going to increase accordingly. So this is the called the other mode of a reproduction, transduction, and this mechanism is considered as generalized. We are going to learn about the specialized mechanism also, but in a next part. First, initially understood about this transduction. Anybody having question? I think you were asking something. Yes. Yeah. Yes. As the virus is getting inside, it going to have the some mechanism in a first. It is going to start to develop certain enzymes. That genetic material, first what are going to do? They are going to cleave the genetic material of our bacteria. For that, they require some restriction enzymes. So first they will introduce, they will start to synthesize it. In so many bacteria, I haven't done or taken more in detail about it. Certain phages, during their packaging, they may lead to certain enzyme will be packed within it. While packaging itself, you are knowing that when you are going to enter or the someone is going to enter, they should have some arms. Otherwise, the public will going to harm themselves. Okay, so they will come with the certain arms. So they are having the arms with them. So they will control and arrest all the class. All the public is going to get arrested. So here, it is just for the bacteria, they are coming and knowing as a protein, nothing else. Okay, protein material is sitting outside and it has transferred the genetic material. Whenever some genetic material is coming from the outer side, bacteriophages do have their, you can say, saving mode or the mechanism, but it is not evolved as compared to the eukaryotic cell. Okay, maybe you are going to learn certain concept related to DNA modifying, DNA repair mechanisms, others are there, or the any genetic material coming from the outer side, it is going to be recognized by the methylation process also. But it is the fraction of time it will may take. But bacteria does not having that much evolved process. So phage is going to control it. It's going to start first what? Protein machinery, and another is the enzymatic machinery is going to start. So it is going to cleave this bacterial genome. As the bacterial genome is disturbed, it is not able to synthesize the basic enzymatic machinery to control our phage. You are knowing that when you are entering, who is the controlling agent over here? Just shoot or control him. Whole class, entire class is going to get covered. It is like any countries do have their parliament and other places are there. Who is the president or prime minister? Just control one person, whole country is yours. Okay, so they have disturbed here genetic material. Cell is not disturbed. Controlling agent is disturbed. Now the second point will start. They will going to utilize for the protein synthesis. So they will completely protein coat will form. And while this process, they will continuously replicate their phage DNA. And phage DNA is going to get replicated and packaging is going on. But by chance in certain phenomena, they will take the genetic material from the bacteria even. And it's going to transfer to some other bacteria. It's called generalized transduction. So I hope it is clear. In exam, it is maybe asked, you have to draw it. You have to draw step-by-step process. In generally, in my papers, I don't know about others, but in biological system, about 50% is marked, especially is given for the figures. If I'm going to evaluate, if it is 8 mark, I'm at least going to give the 4 to 5 marks if it is a properly drawn and labeled. Label diagram, step-by-step process, if it is there, if it is asked in the 4 or 8 marks, I will going to give at least 4 marks for it. Then after for the write-up. Okay, so at least 50% mark, especially in my papers, related to figures will be there. So it is, you have drawn it, just written, don't expect full marks from my end. I will going to give the weightage for both. So till this clear, everybody? Very easy mechanism, right? Now the next step is related to specialized mechanism. Let me move towards it. So initial steps are somewhat more or less similar. Okay, infection. Now the virus has came with some different phenomena here. Okay, sometimes may happens that virus when inject their genetic material, what going to happen? They are going to get integrated in bacterial genome. Oh, here it is here, donor bacterial DNA, just viral DNA. Viral DNA, it's going to bound onto the bacterial gene. It's going to bind with them. It's called the prophage. New terminology we are going to say. Otherwise, in the first generalized transactions, what we have seen, that is going to arrest the bacterial cell. Okay, and they are going to use the bacterial genomic material for their protein synthesis and the DNA synthesis. But here, in initially, it is like that in your class, someone is coming, be a part of the student. Okay, he became the students. He is running all the activities. Afterwards, it may going to arrest the cell. It wait for a while. So what's going to happen? When the cell, it's the genetic material, viral genetic material, it's going to bound onto the bacterial chromosome itself. We have the new terminology called the prophage. We are going to say them as a prophage for a certain period. And after some time, binary fission is going on. Bacterial cell, bacterial genome has got the new virus, viral DNA along with them. That is known to the only to the viral genome. Bacterial genome does not know that I have received something new. And bacterial binary division is going on. So maybe certain generation is goes on. Afterward, that viral genome, when some specific conditions may happen, it is also time independent process. It may be on a second cell divisions or after a generation of generation, it may from this place, prophage, genetic material is going to get excised. The terminology called the excise from their bacterial genome. Now it's became once again independent according to the previously. Okay, it's going to excise and then after going to arrest the bacterial cell. As it is going to arrest the bacterial cell, what's going to happen? It will control it. It's going to digest the bacterial DNA and going to utilize for themselves. That is the specialized mechanism. But during the excision, it has been observed. Here we can see in the next step also. Yeah, here it is saying that during generally in specialized transduction, what is the basic mechanism? It is the ring-like structure. If I'm looking bacterial genome, something has been excised. It will arrest the entire cell, as in happened into previous generalized transduction. But sometime it has been observed during excision, it may take certain additional bacterial genome. It one has to take only. It is like a when they are coming here, it becomes certain became there already a friend. So when he is moving, it is like a prison something. When they are moving, it may take some another guys also along with them. Otherwise, it should be about 100%. It is the genomic material from the virus. It may take around 20 to 30 or 40% additional bacterial part as a genetic material. And what going to happen? It going to packed into this. Because what going to happen? That it is the excision is taken place, just right. So during the excision, it has taken the viral material. Viral material has taken the DNA also from the bacteria, and this is now going to pack into phages, protein material, because phage only knows that I have to pack. Packing you what? The viral genome. But along the viral genome, it is like a trend. Okay, so trend has taken the two additional bogies. So additional bogies now going to bound with it and going to pack. So what going to happen in the next step? It is going to release after a certain time period. Okay, and it is going to infect next one. So generally, as it is a phage, should have a viral genome itself. But along the viral genome, it has acquired genetic material of the bacteria. So it is going to transfer to the next cell. Bacterial DNA along with the viral is there, and it is going to transfer to the next. So here you can see blue to some different color material is being transferred. During the excision, yes, now the major problem is arising that when the viral genome is going to move out of this area, sometime 100% excision for the viral genome, sometime 80% or the 70% is there, and 30% from the bacterial genome also takes place. So all the machinery, genetic material may be driven fully, sometime halfly. It is called the error in a mechanism. But driver is knowing what? Something only this. It is like a when a driver come to know that someone has sit. Okay, but someone has sit with the pet dogs, it going to take. Or some child is taken by the some passenger, it will not going to ask anything. Driver is looking that someone should sit, I will run. So the bacterial protein, phage protein is going to do transfer to the next. So in this process, errorful viral genome has transferred. Error is what? Related to additional bacterial genomic material is been transferred, and it may going to integrate into recipient. The recipient cell has received viral genome along with the bacterial genome. It may be sometime during this process, it may take the whole segment of the virus and the DNA from the bacteria. Sometime it may going to just going to integrate the bacterial genome within it. Other component may be get degraded. It fully going to integrate sometimes going to integrate partly. But by this process, our bacterial cell received something new material in their addition. And then we are going to consider as a reproduction. DNA transfer from one organism to another organism has taken place via any means. Here it is a virus became their carrier, but by any means it has transferred the genetic material, and we are going to consider as a other mode of a DNA transfer. And here we are going to consider it is the process called the transduction. DNA transfer via phages in a bacteria. Okay, but it is not general mechanism. It has the specialized something. Okay, in first step, in generalized transduction, whole bacterial fragment was by chance transferred. In generalized transductions, it was specialized transductions. Bacterial genomic DNA as well as the viral genome mixture has been moved. and we are going to terminalize as a specialized transduction. Okay, so maybe it's the next step similar to it. Now it's going to get integrated within it. Bacteria is going to divide by binary fission, and the cell division is going on, and bacteria has received new some genetic material. The transfer of the genetic material via phages, and this mechanism is considered as a specialized transduction. The small documentary similar to is there.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the GAL gene, near the specific site of insertion remains attached to the phage DNA, and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome, but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
Yeah, so just basic mechanism is there. What I had just told, it is shown in enigmatic form, that during transfer, it is became part of bacterial DNA. That time we are going to term them as a prophage. Prophage, after certain cell division, may excise from their area. Sometimes it may going to excise properly, then there is a no problem. But during the excision, sometimes it may take other part of the bacterial gene. Okay, bacterial DNA is taken up. Sometimes it may leave certain portions onto the previous bacterial genomic material also. And it is when it has phage DNA plus bacterial DNA, but it is having the capacity to get bound onto the your given proteinic code. So this proteinic code is only knowing that I have to transfer this material to next. So it's going to infect the next bacteriophage. As it is going to move inside, it is going to bound once again to the bacterial genomic material. It's going to bound there, and it's going to become a permanent area for them. Because sometimes it may going for, again, it's considered as a prophage once again. But in some cases, it has been observed that while excision, some of the phage genetic material were lost, not 100% has came. So capacity to again excise from the bacterial genome is lost. It has been observed that bacteria has received bacterial genome, viral genome is introduced. But for their process of once again to excise from this area, it has been lost because it has came with a halfway, maybe 30% is lost. And it's now not able to move out of it. It permanently became a bacterial genomic material. It is like that. Now some person has moved from this area to another. He has left certain arms here. He has taken some friends along with him and moved to the next class. And they have integrated over there, but they cannot move out of it because they are now armless. They became a part and parcel of the new component itself. And this way, our bacterial new cell has received genetic content fully new. It was not previously available. And it is got integrated within it. So bacterial genomic size is going to increase and increase. Sometimes it happens that it has taken some good or relatively having some functional gene, or DNA is a functional, or maybe it is non-functional genetic material is taken up, then it became junk for it. Same thing is happened into the higher eukaryotes. So many viruses has infected certain areas. In case of plant, we are knowing, right? So it happened that it's going to get integrated into the genomic material, but they lost the capacity, one has to again excise at the certain place or certain time. But it may happen that it's like certain transposable elements. Have you heard about it? Transposable elements? Okay, no problem. So in future, you are going to learn about it. So many genetic material in this process into virus when they are infecting one to another. During the infection, they have tried to be arrest the cell and to excess, but they have become the part of the genetic material, and the cell division is goes on for their general cell. And the stage where came, they are became part of the genomic material either in a plant or in animal or in other also. And the process is goes on and goes on, and evolution has moved in a way that functional DNA in whole genomic content is less and junk DNA is very high. The additional DNA during this process has increased a lot due to this process, and it is carried by us also, even in higher. The highest, you can say, the genetic diversity, if you are looking, junk DNA is available into the insect. The genomic content of the insect is thousands of times greater than us. But we don't know how it has happened. Maybe it is there, the viruses are being transferred. Maybe insect having the capacity to nectar they are sucking, even they are biting to their animals also. Okay, there are this, if you are knowing, Anopheles and so many mosquitoes are there. Male and female are having the two different phenomena. Males generally are sucking the material from the plants or the oil glands are there, and the female are required additional material for the egg development, so they are biting the animals also, otherwise they cannot develop the matured egg. The insect has got diversified effect, it's affecting the plant and animal. From both, by chance they have received viruses or the genetic material from the various places, and during development, it may happen that it became part and parcel or integrated into sperm and the egg development. In case of insect, also same process is there, and maybe became part, and the junk DNA has increased a lot. So insect having the highest junk DNA compared to the other all in whole kingdom. Any kingdom we are looking, genetic diversity, if you are looking in phylogenetic analysis, if you are looking evolutionary based, then the junk DNA is highly available into only in a insect. But here also in bacteria, maybe beneficial genes has received, sometimes problematic genes also has came, they became sensitive, otherwise it is just junk. It has been given to them, it is of a no use. But we are going to call them as a reproduction. Reason is what? DNA has been transferred from the surrounding by maybe a same organism or another organism. So here phage has participated, and phage has taken their own DNA as well as bacterial DNA is gifted to someone new. And this method we are going to call as a specialized transduction. So I hope it is clear for everyone. Anybody having any questions? I hope it is clear. Yes. Be allowed, I am not able to get you. Yes, there is a chance. This bacterial DNA, it was old. When it has excised, we never know where it is going to inject. Okay, when they have excised, or the first when they have integrated, integration of the viral genome in a DNA is any damn place. It is not controlled by viral genome. So when they excise, they may take a resistance genes. Sometimes some are sensitive genes. Sensitivity means if the such genes are there, cells may die, or their products may be synthesized. It may make them sensitive against a certain process. So maybe beneficial or benedictory, both. beneficial, benedictory, both the thing is possible into this. So it is became sometimes a curse for the bacteria. Okay. Sap, abhisap, banne. Okay, so it is going to get into it. So I hope it's clear. Next step, I think I have already talked previously. Yesterday I was talking. Same we can repeat. Transformation. Here, the bacterial cell is living in the normal cell population. But outer, either DNA plasmid or DNA fragments has came. How they have came? That some cells are dying. When they will die, all the cell content is going to disperse into the media. Any cells, when they die, their cells is going to disperse. So cell wall, enzymatic material, cytoplasm, everything is going to lose out of it. Even their genetic material is going to get fragmented. And this fragmented material, some bacterial cells are ready to take up. It is like that you are moving outside, right? And some items you have received on your way. You may like to take it, sometimes you not. It is like a whenever you're going, garbage is freely available outer side, right? It is extra. When cell died, it is of no use. But if it is recently died, any pigeons or kettles or any animals is recently died, then those who are like taking veg and non-veg, those who are non-veg like her, they may take it. Why to waste? Okay, and another way, if you're moving outer side, somewhere you are finding that apple or the mango trees are there. Mango ripen up and they fall down. You will like to take it. Okay, so there is a no harm. Now the from tree it is fall down, but it is yet you can eat it. So you will take. So when you are moving in your environment, so many things are available. It is like a currently we are having the concrete jungle. Otherwise, you may think like in your forest jungle. In a forest, you will find so many things. Maybe useful, may not be. Or you can use them as an arm even. Certain things can be used as arm or as a medicine. So you are waiting for the good products to be get utilized by you. The same thing is bacteria is doing. Bacteria only knows the best term is what? Their genetic material improvement. For us also, why we are looking for the good partner? It may be some intellectual, having the good salaries, reputation in their surrounding area. This two partner selection is going on between two families, either in a way that by a family members it is selected, or in the new formations we are looking there by their own choice. Okay, it is like arranged or the love marriage we are calling in our terminology. The selection of the character is between the two partners. Maybe some people are associating to helping them. If it is not going good, they may separated or divorce may happen. But the two partners were selected. But in another way, what they are looking for the better, it is the mode of sexual reproduction. Genetic material improvement is required over there. Nothing else. All the organism, all the species are looking for their survival and better genetic material. And that is why the intellectual quotient or the ability to develop the things is increased from first generation to second to next generations. Now you people are highly acquainted with technology as compared to us. During that time, we don't have that. Okay, but during that time, we are having the capacity to handle the so many machineries and instruments very easily that you cannot do manually. You will require the technological aid. So shift has changed. Like that only, genetic improvement. For the bacteria, genetic improvement is the solve of possibility. So what they will do from the surrounding, they will not going to, if by chance they got the garbage, they will throw out. If it is good thing is came, they will take it. The selection is also going to happen like plasmid. By chance genetic material is integrated or genetic material, if it is sometimes observed, it is degraded. It is a garbage. No one like any garbage, waste material. Good material is there, let me take. Okay, by chance if you're moving, you find that some gold or silver necklace is there, you will take, not a problem. You are going to see, okay, nobody is there. It is, I mean, a need. I can sell. Diamond sale is there. Otherwise, I can give to my someone colleagues, he or she will be happy, and life may goes on. Like this only. It may degrade it or may not. Another, when the plasmid is taken up, plasmid while taking, it is also going to get selected. Here, what's going to happen? They are going for the binary division. During the binary division, it has been observed that first, they are looking for the chromosomal material to be get replicated. Afterward, it do have the extra chromosomal DNA that is called the plasmids. Okay, when you are moving from one to another place, right? You are going to select so many items you have taken. Maybe some students are right here only, okay, in a hostel. So during 4 or 5 years or 3 years, they maybe have purchased so many things. It is required for them. If it is like a pillow cover is not good, they are not providing. They like to have the yellow or some colored material is there. They may purchase for so many materials they have purchased. But while moving from here to some place, or if you are in some state itself, and you are moving to some another state, whole family is moving, you are going to sell off so many things. You cannot take every sort of the things, steel dabba or the any material, packaging material, you will sell off. You will take the money and you will going in another place. Like that only, the selection of the genetic material is take place during the replication. Extra chromosomal DNA in a bacterial cell may be 1, 50, 80, 100, enormous number of the diversity has been observed. The bacterial cell will say and check whether this material is useful for me, then only it's going to get duplicated. Otherwise, it's going to get disintegrated. So it is taken by the transformation. It has been acquired, but it is going to not been useful. It is like when you are coming, every teachers are telling so many things, right? What you like, you are going to grab. Other thing is you are going to keep here, thread here. Okay. What you like, you will take in your mind. Otherwise, you are going to note it down. Like that only. Bacteria has received so many things. If they will like, they will keep. Otherwise, they will lose their self and move on. But the method of the getting genetic material from their environment is happened, and genetic material is acquired. That is why it's called the reproduction. Genetic material acquired by any means. Okay, here it is from the surrounding also. So from the surrounding also it is there. Maybe it is useful or not useful. It is not matter, but it has acquired. Then it is called as a reproduction. And this mode of reproduction from the environment, if something is taken, we are going to call them as a transformation. Okay, so transformation with a DNA fragment or the plasmids may happen. So I hope concept is clear to everyone related to all entire space points. Fine. If anybody having questions, you can ask also. So I'm just stopping here. I think our timing is better over. You can put your attendance if it is left into from chat box.